So the talk I'm going to give is pretty straightforward. Basically, just want to go over I guess a few slides talking about the needs of really what represents a comprehensive solution to characterization. And as JJ just said, we have this Q Exactive BioPharma that we just launched at ASMS. This is a pretty neat, advancement in our sort of mainstream platform, the Q Exactive. And so I'll show you what we can do. This is really, I think, the beginning of this, using native LCMS on this new Q Exactive BioPharma system. And then a little bit about how we apply this to the problems and the challenges associated with ADCs. Of course, there's cysteine-linked ADCs and lysine-linked ADCs are the two major categories for ADCs. So just to begin, first things first, biologics. That's antibodies, any of these drugs that we're making. These exist in these micro-heterogeneous mixtures with all of these various permutations. We talk about an antibody, but an antibody is not just one molecule. It's a micro-mixture of many molecules at once. So this is the challenge in characterizing. There's low abundance variance. There's differential forms. So the goal of characterizing, it really can't be had by a single approach. So peptide mapping is really the most sensitive way of going about this, where we can detect sequence variants, post-translational modifications, misincorporations, truncations, all these at the site level. This is the most sensitive way of doing this, but what it doesn't do is solve the problem of the combinations of the abundances of these different forms. So that's where intact is important. These sort of stand as pillars alone. And then in the end, the way to sort of put these two together is with this strategy of top and middle down. So an MS-MS approach as opposed to the intact MS1 only approach. And here, this is basically a way of confirming the structures that we believe to be there. So the essence, I think that the true core of all this is really here at the peptide mapping and the intact analysis. So the intact analysis posed somewhat of a conundrum for us. Orbitraps have been, I would say, year after year, increasing in capability in our own hands and how well we can do intact analysis. So the better that we get at this, the more, that we realize. And so here, what we have, the breakdown prior to ASMS 2016 was we had this one-stop shop mass spectrometer, the Q Exactive, fantastic. This was really designed from the beginning, right out of the gate. The Q Exactive has been an excellent peptide mapping instrument. Orbitrap, high resolution, mass accuracy, quite fast. So this is something that lends itself well to the peptide mapping, as well as top and middle down. The intact analysis, though, we found after the introduction of the Exactive plus EMR, that this really, the Exactive plus EMR was the highest performing for intact analysis. Part of this had to do with the fact that we could access native conditions and perform these intact analyses in another way, which allowed us certain advantages. So I'll get into what that means. The Q Exactive, though, was and is, on this picture at least, limited to denatured intact analysis. So basically the EMR, the Exactive plus EMR has sort of taken on a bit of a cult status. Part of this is because now we can do this native mass spectrometry. And so just to give you an example of how flexible this is, the trapping gas, this is sort of an arbitrary number, this trapping gas pressure setting of 1. One of the parameters that's offering this flexibility allows us to go from denaturing into intact conditions is this trapping gas pressure, where we can really hone in on the collisional cooling for compounds of a certain size. So we can dial this number in and really get the kind of data that we want. As you go from, as we go from this denaturing scenario into using high purity ammonium acetate salt, and as you increase the size of the compounds, you might typically also increase the salt concentration. Maybe this is a non-covalent multi-protein complex. And in this way, we've been able to measure some extremely large things I believe Albert Heck has gone well over 1 million. In this case, Wendy Sandoval, right after the launch, was showing quite near 1 million with this antibody hexamer at 890 kilodaltons. So this is a fantastic mass spectrometer. The question is, how do we get this capability or something that is usable for the core of biopharma back into a Q Exactive? And that's what we did with our Q Exactive BioPharma. So this is really taking the essence of the Exactive EMR and allowing us to now conduct a portion of these native intact analyses on the Q Exactive instrument. So that's what we have here. We're very proud. We have one LC, one mass spec, one software solution for labs of all sizes. This is something for small labs, for big labs. And we have this broken down now into pre-optimized modes. This is the high mass range mode is really the core introduction here with this Q Exactive BioPharma platform. So high mass range mode, HMR, sort of like EMR. HMR now allows us to do intact, antibody, and ADC analyses in both, denaturing and native conditions. We haven't changed anything about the subunit, middle, top, down, nor the peptide mapping. In fact, the hardware hasn't changed. All we've done is develop a new firmware, which has this mode, and it allows us in HMR mode to conduct these experiments. So we haven't broken anything, we've only introduced something new. So with that, I'll just talk a little bit more about high mass range mode and what we can do now in a more automated fashion to bring this to routine biopharma labs. So with our high mass range mode, we have this native size exclusion, which allows us to do native LC-MS. And this is really, I think, the trick to making this happen, usable in a routine fashion. So our MapPak-Sec column is this Thermo size exclusion column that we like to use. In this case, we're using a mobile phase of 50 millimolar ammonium acetate. Purity matters here, so buy the expensive stuff. This is 99.99% metal-free. I would say the results get even better as you go to 999 and so on and so forth. So in this case, you can see size exclusion. What we're doing is we're actually desalting online. We're not getting terribly high resolution chromatography, but this allows us to go from an autosampler just by pressing buttons and spray right into the mass spectrometer like we're used to doing. So this can be very routine. In this case, we're desalting online. We have our antibody, which elutes. Baseline resolved, and then at some point after, we have these buffer salts, the formulating buffer salts that can be completely separated from our antibodies. So the challenge with size exclusion is really secondary interactions. Now, you can add salt, you can change salt. We've really just started scratching the surface here. This is ammonium acetate. I'm sure that we can imagine other conditions for this, but one of the things that we found is that with antibodies in particular, and this isn't really inherent only to size exclusion, but we found that when we have ADCs, when we have these micro mixtures, the elution profiles of the various components are not identical. They're differential elution profiles. So something that we've implemented, we're really proud of, this is something that's unique to Thermo, is this sliding window deconvolution. What this does is, as I'm moving through these pieces of chromatography on the screen, you can see that we're generating different deconvolution component profiles. So the deconvolved spectra change as we move through the chromatogram. That's not new. But what this does is it allows us to get a fair quantitative comparison of everything in this chromatography peak. So basically, I would recommend anytime you're doing chromatography, this is size exclusion, this is reverse phase, where you have different things which may not be eluting at the same time. We're using this sliding window deconvolution. So this is key to when we come back to the antibody, or the ADCs, I'm sorry. So just a little bit about the different flavors of ADCs, and as the flavor of ADC changes, the challenges change as well. So in this case, Etcetris and Kadcyla represent the two commercially available compounds, FDA-approved ADC compounds currently on the market in the United States. These are quite different in their construction. I'll go into the differences here, but basically, Etcetris is a cysteine-linked ADC. Lysine, the Kadcyla is a lysine-linked ADC, and the major difference here with these commercially available compounds is that we've now, in the case of Etcetris, the cysteine-linked, we've swapped the interchain disulfides. These cysteines are the sites of conjugation of our linker drugs. And in the Kadcyla, lysine-linked, of course, we've sort of randomly derivatized all of the external lysines. This is a highly calibrated sort of construction method, and the challenges analytically are pretty different for these two types of ADCs. Now in the future, and sort of in development right now, are these next generation pipeline ADCs. So site engineered ADCs, or sort of glycosylation specific. There are a lot of really clever tricks out there. They're not commercially available yet, but this is maybe going to be easier analytically. We don't know, I hope. So 2 reasons why native mass spec is an native mass spec intact analysis is a powerful addition to a comprehensive ADC characterization strategy. Basically, these are made differently, and the way that native mass spec helps is different for each one. So in this case, the cysteine-linked ADCs, this is, I think, easy to understand. So basically, if you're using denaturing conditions, the light and the heavy chains fall off. We no longer have covalent association between the different chains. If you put it in organic and acid, they fall apart. No surprise. So what is surprising, I think, to a lot of folks that haven't been looking at these types of experiments lately, is that when we move, even for covalently linked compounds, this has an entirely covalently linked structure. But as we move from this denaturing to native conditions, this is in fact this lysine-linked ADC here, as we move towards the native conditions, you can already see that we're getting this nice separation of the charge states. So for very complex mixtures such as ADC, lysine-linked ADCs, this is pretty important. So I'm going to start off talking a little bit about this cysteine-linked ADC, and then we'll see how this applies to our lysine-linked ADC. So brentuximab vedotin, this is the generic name for Etcetris, which is a cysteine-linked ADC. In this case, these drugs are typically made using a linker and a drug, which is conjugated to each other, and then this linker drug is conjugated onto the antibody. So this is sort of the fully saturated form, but there's intermediate forms which also exist. Just to give you some more details here on our linker drug, this VCMMAE, we have an attachment group, a valine citrulline, a spacer, and then this monomethyl arstatin E, a very, very cytotoxic compound. So we can have, in this case, a possible drug-to-antibody ratio of 0 to 8. And of course, for ADC analysis, it's this antibody-drug ratio that we're primarily interested in. And that's why the intact approach is really the only way of doing this. So if we denature our cysteine-linked ADC, this is what we have. So this is a reverse phase analysis. We can see in the reverse phase chromatogram that there are multiple peaks. And as you deconvolve all of this, we can use sliding window to take all of this chromatographic information, generate basically this deconvolved profile. And we see all of these different iterative forms where we're adding more drug, and in this case, adding more drug, these subunits fall off readily, and as we have less and less drug, we have peaks representing that too. So this is a distribution, but to know what the drug-to-antibody distribution is from this data, these data are impossible or very difficult really to gather, and it would be speculative even in the best case. So it's much easier in this case to run native LCMS experiments and understand your average drug-to-antibody ratio. So as previously reported for Etcetris, in this particular compound, the DAR using static nanospray, which has really been this expert level approach, and this is the standard approach for native so far. This has been anywhere from 3.9 to 4.3. We got an average drug-to-antibody ratio of 4.07. We were right on the money. In this case, what we find is with size exclusion, we get a nice elution of our antibody. Then there's our salt peak. We get a profile like this. In this case, we're actually getting a little bit of ejection of light chain. And we've also got a little bit of fragmentation. So I think in this case, I can do better. These are new data. It's not different than the EMR in this case. We have a number of parameters that we can hone in on desolvation. But I'm really happy to report that this is working. These things are staying together, and it's working on a size exclusion column. So it's actually even convenient. Okay, so as we move on, this is on the same instrument now. This is a Q Exactive BioPharma. This is actually a Q Exactive Plus. So we have sample prep, straightforward, reduction alkylation, trypsin digestion, reverse phase gradient. Of course, we get 99% sequence coverage. BioPharma Finder makes it very easy to get both the breadth and the depth of coverage, 99%, but you can see these various pieces that we get either from trypsin miscleavage or in some cases, the various drug loads that we have. So in this case, at the heavy chain hinge region, we have a peptide which is found with 0, 1, or 2 different drugs on it. We're going to look here at this 2-cysteine containing peptide, and in this case, we have, of course, our 0, 1, 2 VC-MMAE linker drug additions. So you can see from this long linker drug that this compound is quite hydrophobic. As you add 1 and then 2 linker drugs, you actually start having some chromatographic challenges. This is a pretty high organic containing reverse phase gradient, and that's really what's required for a compound like this to even come off of the column. So everything eludes here except for the stuff that you might be interested in if we're site localizing a linker drug. Now when it comes to the MSMS interpreting this, so BioPharma Finder does, I think, a very good job, particularly with unmodified peptides. In this case, we're adding a linker drug. This is a very exotic, new and unique, and in the cases of various biopharma labs, these are also highly, well, secret, I guess, at this point, what these linker drugs are. So, does a good job of the unmodified peptide, but it does a reasonable job with, I think, the modified peptide as well. When we look at coverage, now this is, again, both breadth and depth with BioPharma Finder. This is the peptide level. We can see that we get very nice coverage. We're not, in this case, in the 1 and 2 drug containing peptides, we're not hitting on the bound site. So we took a closer look at this, and we found that, in fact, we do generate peptides, well, spectra, sorry, which correspond to really the cleavage of this drug, both at the linker drug side and at the peptide linker drug side as well. So we have diagnostic ions that we can generate, and we can sort of use these moving forward to figure out what other sites might be modified for this cysteine-linked ADC. So to summarize, for the cysteine-linked ADC, we have drug-to-antibody ratio analysis. This is convenient and easy with native mass spec. It's the way to do this if you're looking for a drug-to-antibody ratio for the cysteine-linked ADC. So the hydrophobic linker drugs sometimes require high organic to get these off of columns. This can be a challenge. So the VC-MMA linker drug in this case is labile and collisional activation. So this HCD MSMS spectra, we do have predictable signature ions that we can use to further sort of peruse the various peptides and see if we have any other conjugation sites that we weren't expecting. So for characterization of trastuzumab emtansine, this is the lysine-linked ADC. The use of native mass spec here is a little bit different. The drug itself makes a very different profile, particularly from the intact level. So we have our antibody drug. In this case, it's made differently than the cysteine-linked ADC, where we have a linker, which in one layer is derivatizing, this functional linker is derivatizing onto these lysine, these external lysines. And then we add a drug as a second round of derivatization. So this can be messy. People who have taken proteins and modified them with small molecules, this can be a very messy adventure from the intact level in particular. So different linker drug, and I guess the theme here is that for every ADC, we can take the same antibody and we can modify it with various kinds of linker drugs. You can make a lot of different permutations, and they may have various characteristics that we need to uncover. So in this case, this lysine-linked ADC, we have glycoforms and other modifications that are inherent to the antibody itself. But we also have linker drug forms. In this case, we also might have free linker or linker-only linker-only forms. So this is, I think this is a really cool slide. And so for non-covalent compounds, it's really easy to understand why native MS is important. But for covalently linked compounds, this is a really important slide. So what this does is it shows with denaturing conditions, we produce this spectrum which is incredibly dense. We have a large distribution of species. And in this case, we can see that as we unfold this ADC, it's all covalently linked together. It's not falling apart or anything, but we have so many charge states overlapping that we have mass interferences which prevent us from getting really nice, clean, raw data. Of course, a good analysis starts with great raw data. So in this case, if we can take all of these charge states, reduce them, and move this information to higher M over Z, we get I guess it's what you would call higher peak capacity in the M over Z realm. So we can take this information now and separate this out almost completely. Of course, I think you can see we can push this more. In the Q Exactive BioPharma, we have a mass limit of up to 8,000. So in this case, we're getting nearly no overlap. And this allows a greater separation of co-eluting species. So of course, chromatography is really great for separating different kinds of proteins, but when everything is co-eluting anyway, this is a really nice alternative. So, we have taken a lysine-linked ADC, we have not deglycosylated, we have not modified this in any way, right out of the tube. You can dilute if you like, you can run it. In this case, we can see that the raw data are nearly, at this point, interfering with the neighboring charge states. So the raw data, you can almost read by eye what the drug-to-antibody distribution is. So here, this is a zoom in of the raw data, and now this is the deconvolved data, and you can see just by eye that this thing is very easy. These are confident results. The drug-to-antibody ratio is not a mystery. We can see it. We're happy. In this case, we have a drug-to-antibody ratio of 3.71, and this is exactly what we would expect. To take a closer look, now we can map this back to both the number of drugs as well as the different glycoforms. But we can also see the presence of additional free linker forms. And in this case, we can map nearly every single peak on the screen. And in a mixture of compounds, which exceeds 100, this is pretty cool. So we found that these dead MCC linkers have to do with, we think, maybe an impurity in the beginning. Nonetheless, based on the intact measurements, we were able to take a guess as to what this was. We calculated this monoisotopic mass, and we did peptide mapping with searching for these variable PTMs in BioPharma Finder. The MCC DM1, this is the linker drug, but we also found the linker only. So we looked for this. This assignment was based on our intact data. Of course, we looked for glycans as well. So this was 3 gradients, 60 minutes. We used the smart digest in this case and reduced at the end. We got 100% sequence coverage on both the light and heavy chains. So taking a look just at the glycosylation, we can see that in this case, I took only the top 15 forms. The list went in excess of 30. So we're getting very nice sensitivity here. You can see the reproducibility, particularly for some of these Manos 4, these are not the most highly abundant glycoforms, so we're really happy here, and the reproducibility tells us this is real. As we look at, in particular, at one of these glycosylated forms, you can see that the prediction is actually pretty nice. We're not getting, with HCD, we're not getting cleavage of the glycan itself, or allowing us to site localize necessarily, but we do, de facto, really, we get great sequence coverage on the edges. But we also get these fragments that are diagnostic for the peptide, glycopeptide, and the glycan itself. And as you can see here with this capital Y BioPharma Finder, the GNM, these are these diagnostic glycopeptide and glycan fragments. It's able to sort of do the heavy lifting for you. So this is really cool. This is the zoom out here. We have the lysines. There's 44 in total on this lysine-linked ADC. We could map back the number of lysines that we found with the MCC DM1, the linker drugs attached, 22 in total. And then we had found as well 6 of these linker-only sites. And 3 of those linker-only sites actually had no MCC DM1 forms detected. So I think there's a little bit more work here to follow this up. Just to give you an example of what happens when you have a linker-only form versus a linker drug form. Actually, this linker drug form gives us this really interesting doublet. This is diagnostic, and it's unique to this particular linker drug. Specifically in the drug itself, there's a stereo center. So when you look at the linker-only form, this elutes a little bit early, it's less hydrophobic, but it also elutes in a single peak. And then, just to finish this up, we have HCD fragmentation here of this particular heavy chain hinge peptide, KVEPK, linked at the end with this MCC DM1. So in this case, you can see that it's actually the first K here is what's linked. But you can see that it's the same thing. So With HCD, we do get some labelability here. This compound falls apart in HCD, but we're able to generate both diagnostic fragments for the linker drug peptide, as well as just the drug itself. And there's actually more fragments in here that I didn't label for simplicity. So to summarize, this ADC, lysine-linked ADC, is different than the cysteine-linked ADC. These are very, very heterogeneous. They also call this a random lysine-linked ADC because it's just one round of derivatization and then a hopeful complete round of derivatization afterward. So this is different, but native mass spec nonetheless helps here by preventing mass interferences, greater M over Z separation. So the linker-only species is actually part of what adds to this heterogeneity. And I think that's something to remember with these multi-step derivatization compounds, that there's likely other species that we didn't expect, and so these can be very, very heterogeneous. So the MCC-DM1 linker drug appears as a doublet due to the stereocenter, and HCD fragmentation yields signature ions that allow us to probe deeper and see what else we derivatize. Final summary, ADCs are an important class of emerging cancer therapies, and they're both fragile and complex, and this can be accommodated by native MS. So the biopharma option for both the Q Exactive Plus and the Q Exactive HF, well, now we can do native MS. This is pretty cool. BioPharma Finder is very good software and makes it easy to do these kinds of analyses. And that's it. Thank you.